I am Dr. C.SWAMINATHAN, Assistant Professor in the Department of Microbiology, St. Joseph's College of Arts and Science, Cuddalore. In this video, we are going to discuss about mycoplasma. Now we are going to see about the general features of mycoplasma. Mycoplasma is a small bacteria. The important feature of the mycoplasma is they lack cell wall. Mycoplasma were originally known as PPLO. PPLO stands for pleuropneumonia-like organisms. Mycoplasma are pathogens, commensals, and saprophytes. They are found in human beings, animals, birds, plants, and soil. The pathogenic species of Mycoplasma are Mycoplasma pneumoniae and Mycoplasma genitalium. Mycoplasma pneumoniae causes pneumonia and other respiratory disorders in human beings. Mycoplasma genitalium causes pelvic inflammatory disease in human being. Mycoplasma requires cholesterol for its growth. As they lack the cell wall, they do not have a fixed shape and they are pleomorphic. They can pass through the membrane filter. They possess both DNA and RNA. They reproduce by a process called binary fission. Morphology. Mycoplasma are very small free-living bacteria. They are highly pleomorphic. Shape of mycoplasma varies from granules to long filaments. They are non-motile and non-spore forming. Although gram-negative, they stain better with Zimsaw's method. They reproduce by means of binary fission. Culture. Mycoplasmas are aerobes and facultative anaerobes. The optimum temperature range for growth of mycoplasma is 35 to 37 degrees Celsius. They grow well on enriched media like those containing 20% horse serum or human serum and yeast extract. After incubation, colonies will be developed after 2 to 4 days. If you look at the colony, the center of the colony will be opaque and granular. The colony will be surrounded by a translucent zone giving the appearance of a fried egg. Subcultures are usually done by rubbing the agar block containing the colonies on fresh media. Pathogenesis and clinical features. Mycoplasma causes primary atypical pneumonia and genital infections in human being. Primary atypical pneumonia. Primary atypical pneumonia is also known as Mycoplasma pneumonia. The causative agent is Mycoplasma pneumoniae. It is formally known as Eton agent. Primary atypical pneumonia is a tracheobronchitis. The infection is transmitted from person to person through droplets of nasopharyngeal secretions. Clinical features of primary atypical pneumonia are fever, headache, malaise, and a sore throat. The organism remains in the throat for 2 to 3 months even after recovery. Genital infections. Genital infections are caused by Mycoplasma hominis, Mycoplasma genitalium, and Ureaplasma urealyticum. Mycoplasma species causes non-gonococcal urethritis in human beings. The infection is transmitted through sexual contact. Mycoplasma species are also responsible for causing proctitis, salpingitis, cervicitis, and vaginitis in human beings. Mycoplasma are associated with infertility, abortion, and low birth weight of infants. Laboratory diagnosis. Specimens. For pneumonia, throat swab, nasopharyngeal secretion, and sputum can be collected from the patients. For genital infections, urethral swab, vaginal swab, semen, prostatic secretion, and urine can be collected from the patients. For the culture, specimens are inoculated into special media containing glucose and phenol red and incubated at 37 degrees Celsius. Mycoplasma produces beta hemolysis and the colonies exhibit fried egg appearance. Identification of growth. Growth of mycoplasma can be identified by means of heme adsorption test and a tetrasolium reduction test. Heme adsorption test. Colonies of Mycoplasma on the agar plate are flooded with 0.2% guinea pig erythrocytes prepared in Mycoplasma broth medium. The plate is incubated at 35 degrees Celsius for 30 minutes and then washed with plain Mycoplasma broth medium. A drop of washed fluid is observed under the microscope. If the isolate is Mycoplasma pneumoniae, colonies of Mycoplasma pneumoniae adsorb guinea pig erythrocytes to the surface. Tetrasolium reduction test. Colonies of Mycoplasma on an agar plate are flooded with colorless solution of tetrasolium compound and incubated at 35 degrees Celsius for 1 hour. If it is Mycoplasma pneumoniae, the colonies turn reddish. Mycoplasma other than Mycoplasma pneumoniae do not produce tetrasolium compound. Serological test. Disc diffusion. Discs impregnated with specific antiserum are placed on agar surface inoculated with the Mycoplasma. Development of colonies is inhibited around the disc. Detection of antigen or nucleic acid. Mycoplasma antigens are detected by the direct immunofluorescence test, counter-immunoelectrophoresis, and enzyme immunoassay. Specific DNA in the respiratory secretion is detected by PCR. Detection of antibody. Antibodies can be detected by means of heme agglutination inhibition test, Streptococcus Mg agglutination test, cold agglutination test, complement fixation test, and enzyme immunoassay. Coming to the treatment, Mycoplasma are highly susceptible to tetracycline and erythromycin. They are resistant to penicillin and cephalosporins. Thank you very much for watching this video. Thank you.